questions that we have prepared beforehand, and if the members want to ask questions themselves, they'll ping me, and then they'll unmute themselves. So
Your mic is coming in and out. Can you hear me okay?
Kind of broken.
yeah, hold on. I think it might be my sensitivity settings. Hold on. All right, how's that?
Much better.
Okay. So, I was trying to say that we have a couple of questions that we prepared beforehand, and if people want to ask their own questions, they're gonna let me know, and then they're gonna unmute themselves and talk.
Great.
Uh, but first, before we ask any questions, why don't you give a little introduction to about yourself and like kind of your journey in the science of myrmecology, especially for some of the members who don't know as much of your story as others. So, yeah.
Well, I didn't always know I wanted to study ants. In fact, all I knew growing up is that I wanted to do work outside the forest, place. And I actually think you're going to have to mute yourself
Oh
while I
yeah,
speak.
sorry.
There we go. Now I can speak. And I always wanted to find a job or do science outside, and I decided I wanted to work in the tropics. That's all I knew. And I thought I'd study plants. I prepared myself to study plants. Then, as a basically a freshman undergraduate, I decided that I wanted to go to Panama and work, learn about the tropics. I went there as a botanist. As soon as I got into a rainforest, all I saw was the ants. And even though I was working on plants and collecting plants, I just couldn't stop collecting the ants and asking about the ants. If you've ever been into a rainforest, it's the leaf-litter ants that you see, army ants that you see, but if you really start looking, ants are everywhere. They're just like almost everywhere you look, you see ants. And to me, that was super exciting, and also super exciting that nobody could tell me anything about it. So over that 9 months I spent on this tropical island in Panama, I switched from studying plants completely studying ants. In fact, my first papers I published that year were on plants, and then I studied relationships between ants and plants. And by the end of that year, I was just doing research. And it's some of those questions I began asking at that point, the same questions that have driven my research around the world, wherever I go. And I was working with botanists at the time who were much further ahead than we were in the ant world. So their questions were very exciting. I felt we could do the same thing with ants. And of course, there's birders that always come to the tropics, and they had great things about birds that they could do. And I wanted to do the same thing with ants. So since the beginning, I've been really trying to bring ants to the same level as you can do with birds and plants. That is, go to a place, find out what's there, and understand how those ants change across the landscape. After that year in Panama, that's what I began to do. Now, it's kind of confusing. This is kind of what botanists did already, what bird people did already. And science was moving on to this kind of real experimental design and ecological testing. I wanted to do basically 19th century science. And going to grad school was actually a challenge because what I wanted to do was not science. And it was basically required move ants to where you could do what would be more fashionable types of science research. And I just had to figure out a way to do that. And it was challenging, but I found a way, and other people are finding ways too. And it's a very exciting life to be out traveling the world anywhere, especially the tropics, where you can go and be almost the first person to go and document. That was my brief intro.
Okay. All right. Is my mic being a little bit better now?
Yes, yeah.
Okay. So The first question we have, which you kind of answered a little bit, but how were you drawn to the study of ants?
I guess it didn't really decide it had to be ants until I went to the tropics. Temperate ants are great. They are amazing ants. We have, stigmatoma in California. We have Proceradium just outside my house. But it really was going to the tropics and seeing them as a collective force of nature and in that incredible diversity. So it was that moment that ever changed my research direction. It was experience, and I hope you all would have the chance one day to experience it.
All right. Our next question is, what does an average day at your job look like?
Well, like any job, I have to wear multiple hats. I'm actually working in the collection. It's basically like working in a store, a grocery store. You're a clerk. You're filing and organizing and labeling and databasing. I could be doing the same thing maybe in a FedEx office. That's one of the elements of working in entomology, is that you have this kind of extra level of organization that has to happen. Ornithologists don't collect as much as entomologists. The whales, you don't collect almost anything. Insects, you collect a lot of stuff. So you can't get over that, kind of rise above that mass of organization unless you're super organized. And as an entomologist, you got to be. And those are the wide collections. In fact, one of our collections at the California Academy of Sciences, where I'm based, is now one of the largest ant collections in the world. I was able to organize massive collecting, and processing, and labeling, and databasing, and imaging to make that happen. So you can't ignore. You got to be a clerk. That's one hat. The other hat is when you go out into the field. There, it's a completely different hat. That requires a whole different skill set of being able to handle travel and in places that are unknown to you. So you have to figure out how to actually live there, survive there, do logistics there, manage how to get the permits in that country, work with the locals, figure out the language barriers, and get to the forest. A lot of this forest is not accessible. You have to manage to get there, and you have to get out. And the field work isn't done. The specimens are back on your desk. There's a lot of traps that can happen in transport and shipping. Ideally, a biologist would be given a diplomatic passport, travel anywhere and come back with it. We're not. We're almost treated as criminals at times. We're traveling. And even when you come back in the U.S., even though you have all the permits, even though you know the laws in and out, still, when you come through that door and declare that you're bringing scientific specimens into the country, they're going to treat you like a criminal at times, depending on who you get. I mean, it's going to be very disconcerting. And that's one hat. And then the other hat is example right now. All I can do is write some of those papers that I haven't written yet. I'm sitting, doing research and connecting the vast data that I've already collected and trying to put that into paper. That's the other hat, is being a writer, because there's a difference between being interested in ants and being a scientist. The scientist is somebody that takes that interest, that's the drive, to get you up in the morning and to see it through, and takes it to the point where you can actually synthesize, analyze, and put it out in the world. Otherwise, it's just an interest, a hobby in a sense. There's nothing wrong with that. There's a distinction between jumping to the point where you can actually make a contribution to that scientific community. Now, for the audience, maybe there are some people who aren't scientists and they don't know what that really is, but I just want to take a moment to explain that for a kind of way. So you maybe are already a scientist. You're not one that actually publishes. You're a scientist. All you have to do is ask a question. We're all probably asking questions all the time about ants. That's the first step. And some of those questions are like throwing out an idea outside. Like, imagine you're standing on this island and idea is just like, hey, about this biology or life history or why it's doing this. It's like throwing out a rock way out that island. And next step is after you have a result, see if that result kind of connect back to the island somehow. I mean, using current research and ideas and theories. Sometimes it doesn't happen, so you just let that rock sink. Other times you can build a bridge back to where you are. That's the artistic part of being a scientist, is making those connections. And often now it's best done as a team. I look at all the people that are in this club as potential players in that team, because you're all asking questions, you're all observing things. That's how this kind of collective knowledge can rise faster. In fact, birders have been doing this for years, right? And I'm glad to see that the ant community is coming together, forming these clubs, and it will really be the future of what we know about ants. It's like it is for birds now. It'll be through these types of clubs. Actually, don't just show an interest, but actually get that data and results out in some form. And it's really not clear yet how that's going to happen. I hope you can figure that out.
Okay. The next question is, what are 3 tips you have for students who plan to study entomology?
Studying entomology is the life, right? Happy life, because you're never done. And it's amazing how long it takes to make contributions, because look at me, I've been studying ants. First went to Madagascar in 1992, and I'm still describing the new species I collected over those first few years in Madagascar. We've described a lot, but it just takes a long time. And do that now as an entomologist. And if your interest is in species discovery, which is just one type of entomology, takes more, I would want to say, a combination of artistic and analytical skills. And probably I prepared myself when I was in school. A lot of the data is now global. A lot of the research being done now requires programming. So requirement now to start as an undergraduate, or especially for grad school, you got to know at least Python, or if you know Python and R, and are already the expert in your group, basically. You go to grad school doing research on diversity and discovery, you'll be so busy focusing on your particular research question, you won't have time to actually become the expert. So spend time as an undergraduate, like with these clubs, really getting a solid background in natural history, observation, also learning how to collect. A lot of courses that are out there that can teach you this. There's a few in the ants world, and some for undergraduates. There's one now, kind of the ant course at the Southwest Research Station. I teach one at UC Berkeley. If you're in the Bay Area, you're welcome to take it. We have a lot of undergraduates and even high school students who have taken it. And of course, when you do start grad school, make sure you take ant course. And this year, we just postponed the course. It was going to be taught in Cameroon. And we've already scheduled one for next year for Vietnam. I don't know what's going to happen, if I'm going to switch it for Cameroon or go ahead with Vietnam. That's another course. And don't confuse your interest with going to grad school. Grad school is a way to actually do research and get a degree. Your interest is for life. Don't compromise your interest. Make sure you're going to grad school and something you care about.
Okay. The next question is, what taxa is your favorite and why?
Not taxa. That's probably what ant species maybe, or what group of ants?
Yes.
Well, I'm really attracted to fine ants, and those happen to be often those that live in subterranean habitats, or just tip of the leaf litter layer. So in ants, there's three main categories of where ants live. There's the arboreal community. That's really anything above ground, all the way up to the tip of the canopy. It's about a third of all species, like in a rainforest, or even 25% at least, rainforest. And we used to think that's where we'd find the greatest diversity of undiscovered species. As it turns out, that's not true. We basically know most of the arboreal ants already. It's really the subterranean and leaf litter area is where we're going to find the greatest unknowns. We have no idea what's in the ground because it's such a hard thing to find ants in. You got to dig, got to be very patient. Often go to the forest, almost lay down and sit there for 8 hours just digging and finding things. And maybe it's the rewards are incredible. It's a bit like gambling, I guess. Digging, and occasionally you have this incredible win. Find a leptonella, you find a stigmatome or a prostradium. It's exciting, and to me, that's what drives my collecting is through the potential for discovery. And subterranean ants need a lot more invention towards how to collect effectively a subterranean ant. We're working on it. We have a lot of tools for testing. Some crazy ones, some really crazy ones, but we'll publish some of them, I hope. You can all laugh at them.
All right. The next question is, what are misconceptions and debates you often run into within the study of ants?
What types of debates? The question you right now are going on in the ant world? That's
Yeah.
something?
Essentially, yeah. So, like, what debates or misconceptions do people often run into?
Well, there's definitely debates, and there's also public misconceptions about ants all the time. A little bit about both of those. My field at times is taxonomy, so I can talk a little bit about that. And there's this, I think, misconception that it's kind of relatively easy to describe a species, and that really don't have to much to actually get going, describe a species. You find something, you go online, you don't find any picture like it, and you go, gotta be a new species. Maybe I'll describe it. This happens all the time. And me as a taxonomist, when I get a paper like this, as an editor of a journal, I always think, I wonder if these are the same people that want to like nuclear physics on the weekend, like that. Taxonomy is a very old field, 250 years, and some of the language around it has been very generalized. We have a code of zoological nomenclature that's written in a very kind of simple language, and it makes it look like it's actually hard. However, describing a species is much more work than you could imagine. This is why. I have to think about that what we're describing is not a car. It's actually a living, changing organism that has a past and relationships. And those are unknown if you just have one specimen. You have to collect lots of specimens over a wide range. Understand the limits of variation that you can actually describe. What are those distinct styles of variation? So look at it this way. So if you were going to like talk about North America or the United States, you're going to describe it to somebody, the first thing you would do is kind of draw it on outline and then kind of major cities on that. And think of those as the genera. Then you might want to draw some more smaller cities in there and those are maybe some species groups that are very, very distinct and so forth. We have most of those done already. So the hard part now is distinguishing which of those villages are also species or not. So that requires looking at all the villages around those cities and figuring out how if they're all the same or different. And it's very, very tedious. All these publications where they describe one species often adequate because they're not taking into account other species that are similar. So that's just one idea. Another misconception I think a lot of people have is that it's possible to identify all the ants like North America. To me, even as a taxonomist, I'm a bit embarrassed that our field is so unadvanced that still to this very day cannot create a key ant species of California. There are too many unknowns that we do need more taxonomy. We don't need that one species description. We need a revision of gamma, for example, of species of chromatogaster. We need all those hard tasks completed. And it's unfortunate we don't have that done yet, and I don't see anybody working on it.
Okay. I'm gonna skip one of the questions that I asked and go to Austin's question, which is, what are the steps to take to getting a career in myrmecology?
Oh, that's actually interesting. Myrmecology is a science that kind of uses ants. So ants have entered into so many different fields of science. You could study the brains of ants, neuroscience. You could study the venom of ants, it's toxicology, and you could study the bacteria and gut biomes of ants, the epidemiology of Wolbachia, and symbionts of ants. You could study the behavior, study the evolution. So there's many, many areas. You can't study it all. The first step is to figure out what angle do you want to enter into the world of ants. I've come in through the angle of diversity, evolution, taxonomy. That's what I chose to do because I felt we couldn't do much else unless we at least understood what ants are out there. Now I realized it's much more difficult than I thought, and it's going to take some time. But once you do that, you're just going to have to start at the same time start learning about ants in general, and also learning the area of your expertise, neurobiology, taxonomy, and so forth. So combine those two things. And the younger you are, the more you could focus on the natural history and diversity and identification, because you don't need any degree to do that. You just have to go out and start collecting, networks, talking to people, and learning how to collect. And you could learn. I always thought I should create more videos on this online because that's really a fundamental to keeping your own collection in this. That's a resource your life, and it's a resource for the world, and there's no reason why you can't start contributing immediately in that. But you can no longer go out and collect birds, right? You're not going to go out and do that. Most people almost frown on collecting butterflies these days. You can collect ants almost anywhere, and nobody cares because they're perceived as a pest. And I'm not going to change people's perceptions on that because it's really only thanks to that allows us to out and be able to collect these. As soon as people start treating ants like they do butterflies and birds, we will have a hard.
I think you cut out a little bit there.
Did I?
Yeah.
Can repeat something again? I'm not sure what happened.
Uh, well, basically, you were just saying how, um, as soon as they take away, basically, you were just saying, as soon as they take away the perception that they're pests, and I was assuming that you were trying to say, uh, then it becomes harder to collect them.
Yes. I was I was just thanking that ants at one level are perceived by the public as a pest. It gives us the freedom to still go out anywhere and help them. Of course, at certain places you have to get a permit. Almost in all cases, it's easier to do it for ants than it is for butterflies, definitely for birds.
Yeah. Okay. So next question is: out of all the species you discovered, which one is your favorite?
Oh, I think that some of the stigmatoma, the Didymerma, the Dracula ants, are my favorites. Also, there's so many. I think it's the ones that were the hardest to collect. There's this an endemic genus in Madagascar that on certain mountains there's a species that is only on cliff bank. Imagine going through a forest and finding a stream that's cut away a bank on that cliff bank, or there's been a erosion and land broke away. There's a cliff bank there. There's this ant that forms hollow in the cliff bank and then forms like an ear on the outside in the clay. You see this from the outside, you think somebody was doing pottery, and they form this beautiful like ear that's really smooth and beautiful. And there's these yellow ants that are sitting around it. They look just like a phoenogaster. In fact, I thought they were a phoenogaster, but as it turns out, it's an endemic genus to Madagascar, completely unrelated to phoenogaster. And it seemed like a very silly ant. There's only like 12 to 20 workers in the colony. And you look in the little hole, and all the larvae is just sitting in there. And it's like under no protection. You're like, what is going on here? There's a lot of kind of army ants, the Serapakines, the Dorlines, Madagascar, that would eat all those larvae. And we watched them, and as soon as a predator ant, which is the number one enemy of an ant is another ant, come and make its way to the lip of these ants would run out, grab the individual, and jump off with it. We called them the hero ants. Each ant would jump off and save the colony from this potential predator. And it was just amazing to watch this. They're small colonies, so they only had like 20 workers total, but they would keep sending them off to protect the colony. It seems to work. So that's another beautiful ant that's found up in the mountains in northwestern Madagascar.
And have those ants been described yet, or are they still undescribed?
No, it's described as Malagidris is the genus. You can find beautiful pictures on them, and I included Google hero ant. Madagascar, it might show up. And we did some other tests on the hypotheses, like, well, maybe the ear is actually there for aeration because you can't really get any oxygen through the clay. We tested all these ideas, and it seems to be best served as a way to limit predators only through one entrance, and then to have these hero ants save the day.
Fascinating. I just looked them up. All right. I think we'll go to the next question, which is: if you weren't going to become a myrmecologist, what field would you go in, like in general?
Well, I was already in botany, and I didn't with ants. It would be another insect group, or it could have been spiders, I guess, or I could have kept with ants, I guess. But it would have been for sure something in the tropics with incredible diversity. I find rainforest most incredible habitat. It's really shocking how night and day it is. You know there's a rainforest out there, like in Madagascar. You're hiking through barren grassland for day after day, and you can see the forest, and then you're getting closer to the forest, and the whole time you're just walking through secondary grassland composed of mostly invasive species, and the second you walk into that forest, everything you see could potentially be a new species, something never observed before. And it's really a privilege to have those experiences. I hope you all will have the chance to do that.
Okay. Um, I think the other members will want to ask some questions soon, so I'll go to this, and then if they have any questions, they're free to ask them. Uh, but I'll choose the next question. We'll do let's see. What are the most memorable experiences from your time in Africa?
Africa. But in Madagascar, I did do a lot of work in the Congo Basin, and I found the Congo was the first area I worked in. And in Madagascar, I traveled on the road like for 6 months at a time. So I had the infrastructure to do that, a vehicle, a team. And a lot of my experiences there were about vehicle recovery. We had to get the vehicles to a forest and back again. Well, in Africa, I didn't have any of that infrastructure. It was basically most of my time was spent in forest, working there, and I didn't have the option to keep moving. And sometimes I fell ill, and one time I was almost dying from malaria. Even though I had medication, it was resistant to it, and I ended up getting help from the pygmy village, and I was recovering there, and I actually spent 6 weeks living with this group of pygmies and flock of pygmies, and it was one of the most eye-opening experiences of my life to be able to witness their ways and understand how they lived and not speak a word of their language. And to me, as a young student of ants, to me, this was a, a dream to be able to do that. And I was really just unaware of that came with the package of becoming an ant biologist.
Okay. Well, I think Mama June wanted to ask a question, but I'm not sure. So if you want to, Nolan, go ahead. Um, okay.
Yeah, I did. So one of the questions that I had for you is we see a lot, like I see a lot in the community, kind of with species that are polymorphic. We see people saying like, oh, they have super majors, or that's a minor, or like just kind of arbitrarily assigning like different castes to certain workers. But like, what's the, I guess, the proper way to go about doing that?
That is a really good question. It's something I've wrestled with recently. I'm sure you're all aware with AntWeb, and I, my goal has always been to change AntWeb, improve AntWeb for identification. One of the problems with if you have a major worker, the species like Phytoli, it's pretty clear you have minors and majors. No problem. It could be a problem because some people might say minors and soldiers, I guess. But anyway, I wanted to make a system on AntWeb where you could choose only see all the majors, only see all the males, or only see all the queens. Only see the queens that don't have wings. I had to divide all three species into a caste, every image into a caste. I had to do it by hand. I had to sort all 54,000 imaged specimens on AntWeb caste, develop a system was robust enough to work across all ants. And this is what I learned. That there's a practical way to describe ant size based on what you see, based on size. And then there's a development pathway, which you could also call those resulting sizes. For example, some of the larger ants are actually on their way to be a queen, but they stop short. They're actually queens that are just not queens yet. And people suggest those types of large ants are ones that have ocelli. Ants have ocelli, those three sets of eyes that are on the vertex of the ant head. For example, some Carabara soldiers have ocelli. So that would suggest that these aren't workers at all, but they're actually queen-like. Therefore, we can't call those major workers because they're actually not workers at all. They're the queen lineage. We need a new name for them. So that's when people start saying, okay, we need to call them soldiers because they're actually a different developmental pathway. While Idoli, it's just a large worker. We can call large workers majors. In the literature, you'll find that people like Christian Peters will say that if it's a developmental pathway on the way to a queen, but it's not a queen, we'll call those soldiers. And if it's a worker pathway and they're just a larger worker that kept growing, call those majors. So I couldn't follow that in dividing Antweb images up, so I decided to ignore that question. I just wanted to figure out how to actually all between worker and something larger. And for some groups like Phaidoli, you have some divisions that are very clear. So you can call them minors and majors. Others, like Camponotus, for example, where you have polymorphic majors, becomes really hard to distinguish at times. And others, where you have majors that are actually are so similar to workers at times, it's hard even to tell if they are majors or not. I use simply a size class division. Phalaenopsis is another example. And out of group, for example, there are 50 size classes, and ADA the same thing. It's really irrelevant, but if you take a practical point of view, sometimes identification, your keys, you're going to have to actually decide if you're dealing with a major or a minor. You have to look at it relatively. So if you got a good colony series, identification is often easier to take some of the larger workers, use those in the key, even though there may be polymorphic. And even though in the middle ground, key might or might not work for those, it's safer to use the larger ones.
So then, in cases of I think there's a few Phaidoli in the southwest, like Phaidoli Rhea, where we've kind of designated that they do have, at least from the literature I've seen, that they do have actual I think the term is super majors that they use in the papers that I read, and there's a few. And in the case of Phaidoli Rhea, they use the super majors to block the nest entrance during Neva Myrmex raids, right? And so my question kind of is more to, I guess a follow-up question that I'm asking is, when do we decide, okay, this is a major, but now when it hits, this point, it's a super major, or you know what I mean? That's kind of.
It's going to be very different for every genus, and there's a tradition. So in Phaidoli, normally you only have two classes, minor and major. Very rarely you actually find polymorphism or different classes within majors. So that's when they're like, oh, we got to give a new name here because this is different. So it's true. In Rhea, you have different sizes of majors. That's all it is. Different sizes of majors. And you have different sizes of majors. And just like all through ants, it's kind of pushing differences in size is what they do for a living. And it's the larger ants that will do different things than the smaller ones. And that's probably why there's this push, Rhea, to actually protect itself from other Myrmecologists or other ants that are coming in and raiding young, I guess, the army ants even. A beautiful ant to see. Have you collected those before?
No, I haven't
Oh,
had the chance. I'm
oh,
very
it's wonderful. It's actually very easy to find near Portal, Arizona. So if you ever go down to Portal and take that class down there, you can just drive off
okay.
and on Foothill Road and up to where you see a little ridge, um, rocky outcrops, and just walk up to those rocky outcrops, and you'll see them on the ridge line usually along those
Oh
little
wow.
outcrops there.
Yeah, it's a dream of mine to make it down there. So I'm hoping to go to University of Florida for my like entomology degree and then graduate school in Arizona. So I'm very excited about that.
By Florida.
I am just because I think I was looking at it with my college counselor. I go to community college right now, and it just kind of lines up well with what like transferring and everything. And then I also kind of like, I would like the opportunity to look at the ants in Florida and stuff. Kind of the same thing going on with Southern Arizona. So that's
That's
why.
like
I
the
mean, maybe
tropics.
maybe there's better schools out there. I don't know.
No, that's fine. I just saw that you're in Illinois, and there's a great school, Champaign, um,
Oh yeah.
and it's a great place to be an undergraduate. They have a great entomology department. The head of the department, May Birnbaum, is fantastic, and there's actually a wonderful ant researcher there, Andy Suarez. He has wonderful students. If it's something to think about, even as an undergraduate, actually shop around. It's the best school for that,
Okay.
and go down there and visit with Andy and tour the department.
Okay. I will. Thank you.
I think Tom has a question to ask next.
Yes, I do. First, I want to say hi, Brian. I'm a very big fan of your work. I really admire what you do.
Thank
And
you,
my
Tom.
question is, how can I, as a amateur myrmecologist, help with myrmecology in the sense that because I'm collecting in my local area, and so what I want to do is I want to be able to, when I get enough findings, I want to be able to help get that information out, get that data out to the world. Is there any advice on how I can do that?
Yes, I'd really like you're doing that, doing your own collecting, amassing your own expertise on your local ant community. I mean, that's where your real kind of growth and understanding will happen. It's actually being able to actually know fauna. It opens up whole other possibilities of observations. They change over time. There's so many. It opens you up to so many cool things you can do. You don't have to go to the tropics, but you can do so many cool things once you master the ants around you. Like when you say, "I know the ant," and you can in the UK. There's only 44 species in the UK, I guess. It's possible to do that. So I would, as a hierarchy of ways you can. And when I say contribute, I'm thinking of more the kind of the global information about it. There's information that's in digital form, information that's in kind of written form. So you can publish, but that's a high bar. Usually you can make observations and team up with somebody, then publish that. You can actually start on the nuts and bolts of helping gather data about ants. And I spent a lot of time doing that. I can talk, and that's why I think I can talk about it. And that is that I've created AntWeb, which is for the last years we to amass knowledge about ants. And also AntCat, which is the literature, names, and the taxonomic history of the ants. And I can talk about those programs in detail if you have questions, and I'm always open to how to change them to serve a wider audience. They're my ideas, and I'm welcome to suggestions. But what surprises me is that there's only relatively few people that actually contribute to those types of resources. So everybody uses, which is always, and I think amateurs maybe. Oh, I think they can grow and improve now because amateurs can help with them. It could be, for example, AntWeb takes certain types of data. One, it records images. So if you have just pictures of ants, you can actually contribute those to AntWeb yourself. You can go to a species page and upload an image. You don't have to do anything except the login. But to actually become a master of specimen data, you have to learn a few things. You have to learn the kind of standard that we've established in the ant world on specimen data. And it takes a little bit of effort, but that hard. And then you learn how to record data, and it actually makes you better about recording the data when you go out for your own interest, because you've learned how to do it in a systematic way. That data, your specimens, your collection, your label information with your name on it can get onto AntWeb. That's distribution data that everybody can see and everybody knows about. And that's how you can contribute to this kind of global effort. And we, by surprise, to be honest, even taxonomists don't give much emphasis to specimens. That's a shame. Yes, it takes a little bit of work to organize. Yes, you have to actually update identifications all the time. It's really raw data for what we know about the distribution. And AntWeb is trying to go into museums and record that data. That's what we do. But still, people who work with specimens often don't contribute that data to AntWeb, because they just kind of take data from the specimens, publish data, and then about the specimen just in a drawer. If you're interested in that, I'm welcome. You can email me sometime and then send you a template and start recording data. And as you become an expert, you could be monitoring them, looking at they change in their abundance throughout the seasons and years. Do the males get produced? Do they produce queens? All this kind of local life history metrics that need to be recorded about species.
Yeah, that's something I know is very true, especially because I have a friend who's an amateur astronomer, and in terms of astronomy, it's often the amateurs that make the most contributions. So I'm really looking forward to being able to contribute to AntWeb potentially, record my local ants, and help you guys out, because ants have really changed my life, and I really admire all the people that work with them.
We have one of the best there in England. I don't know where you're at, but if you ever go to the Isle of Wight, Barry Bolton lives, the master himself.
Oh, yeah, I haven't been there yet, but I'll definitely be going sometime as I go along my journey in myrmecology.
I had another question, like kind of a suggestion that I want to know like the validity of. But so for AntWeb, I was wondering if there's like, is there a way for like, you mentioned being able to upload a picture on a species page, but is there a way to like put an observation on AntWeb, like that we have observed, as like, or like an amateur observation or something like that?
So you have an observation. It's like a specimen record without the specimen. And we do already have some observation records in Ant. They're actually from a published observation record, so in a sense they're literature records, but we don't have specimens. They're treated as observation records. They're from a bunch of studies done in Brazil. And we found out, though, that over time they became problematic because we could not go back and change their identification. For example, for observations of Pachycondyla 1, so country, South America. Well, now half of those Pachycondyla became 9 different genera. We don't know what we're going to do with that Pachycondyla. Is it still a Pachycondyla? We couldn't go back and find the actual specimen and then go, oh no, now it's a Pseudoponera, and we need to change it. Couldn't do that. So for observations record, we now require some, like an image of some sort. Then now we're all of a sudden, it's like iNaturalist, where maybe for observations, it would be better to send it to iNaturalist then a certain quality of observations from iNat go into AntWeb. iNaturalist is from the California Academy of Sciences, and their office is just next door to mine. And we've always talked about figuring out a way to actually better go back and forth. We use our ant classification for iNaturalist. We haven't taken data yet, and I haven't figured out what cut to do to get that. So I think for observation data, because the system often requires a photo, it may be easier to do that through iNaturalist initially, and have it travel through time and be, identified and so forth, and then move it over to AntWeb as a specimen record from iNaturalist.
Well, I was like thinking specifically more like if you had a specimen or something and you've already submitted that, but like with the specimen, you also recorded some observations of like behavior. Like for example,
Oh,
if you this might be an AntWiki thing, better to post on AntWiki or something, but basically if you go to like half of the species of Lasius within the Acanthomyops group have like no relationships between aphids reported. But if you flip like any rock, you can pretty much find Lasius with aphids or scales. Um, uh,
so anybody can add observations for a species on AntWeb. On the overview page, there's room there for you to make observations. Not very many people do, but Jack Longino goes crazy, and he's has a lot of observations for like most of the Costa Rica species. And you can also make observations on your specimens. So if you submit data to AntWeb, there's a specimen notes field that you can just fill up books on. It's not limited. You can write as much as you want, and that gets uploaded. And other naturalists really get into that with their observations of what the ants were doing when they collected them, and that's a specimen record. That's great because that information is tagged to actual specimens. The taxonomy can keep changing of those specimens. If it wasn't identified the species in the beginning, then it gets identified, and those notes just travel along with it. In addition, you can actually make notes on somebody else's specimen. You can actually go in and have a comment on somebody else's specimens, and I can help you find that out. And if those aren't clear, those are ways that we can improve AntWeb to make it.
Another question I had specifically about AntWeb is like, for example, a lot of people in the U.S. they use AntWeb all the time, so they like know a lot about their regional ant fauna. And oftentimes they have like identified species which aren't recorded in AntWeb. But like when you try to reach out to the like page curators, half the time it takes them 3 months to respond, and then they're like for example, I reached out to the the page curators for the Colorado, and the thing like one of them still hasn't gotten back to me, and like the other one, David Lubertazzi, he said that you need like more concrete evidence or whatnot. So like for example, if we have a specimen like of Dory Myrmex insanus, which that one I know they're like kind of messy taxonomy, so you don't want like most people don't want to have to classify them as that, but or like a specimen of uh yeah, that's one of the main things. But like if you have a specimen uh and like for example the Denver Museum doesn't have any ant specimens, but we want to send a specimen somewhere, like where should we send it so that it gets
If you send a specimen that's a new record at the state level, that's important. You should send it to like the California Academy of Sciences. A lab will image it, we'll database it, it'll come up on AntWeb, and the community will be able to see the image. So we image all kind of new records. Example. Now you're right that Dory Myrmex insanus is difficult to identify. However, just now there's a woman that's in the revision of that, a Phil Ward student. So specimens, if they were sent to the California Academy of Sciences, would then be included in the revision. She comes down and uses our specimens. It would be properly identified and verified. So it's true. It can be tricky. It can be complicated to identify at the species level. Now, sometimes you would think, my gosh, this is just a Pogoda myrmex. It should be really easy to identify it. Now, they're difficult to identify.
So if we do have a specimen and we want to send it to you, what kind of format should we like send it in? Should we pin it, or like if we use alcohol, like what percentage and what type, that kind of thing?
Plants, the best way to preserve them for long term and for shipping is, like at least 25%, but I use 95% alcohol. And you have to have full data. Ideally, you also have that in kind of digital form. But when you send alcohol specimens, that means there's a lag. We have to kind of mount them, label them, and then that's just an extra step that has to happen. It's even better if you know how to mount them, know how to label them, send it already mounted, and we can just start imaging it right away and send the data electronically. We can digitize the information. So we look at the ants first that are already mounted.
I don't know.
Those that are in alcohol, kind of getting the key to get mounted up and so forth.
Do you have like a guide to properly mounting and like labeling the specimens? I know you sent me that like the 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 recording observations, like the spreadsheet for like specimen data, but as far as like printing a label and cutting the, or like getting the mount the right size, do you have like recommendations for that kind of thing?
Yes, and we teach that in great detail during our courses. And it's an embarrassment I haven't jumped on the YouTube, possibilities and done some videos. It's clear we could do some very cool videos. I think I tried initially, but a lot of this stuff happens between a normal kind of video, and then all of a sudden you have to go in the microscope and video. And actually, as it turns out, it's hard to do the microscope video work and have depth of field and actually show people something. So at one point, we should figure out how to do this, because it's clear if we're going to have more amateurs involved, we need to have very simple how-to videos. And we might as well have people doing it as best as a scientist right away, and there's no reason why not. We can train people in our courses, train people in my lab. We have the only other way right now is to go to a lab, do an internship, and learn how to do it. That's how I think everybody who's an ant person right now has learned. They've taken ant.
I had a question. So, in the Midwest, I think it's really only the Midwest right now, we have two species of ant. We have Tetramorium immigrans, as I'm sure you're aware of, and we also have Tetramorium, I believe it's Sushime. And
In the
so,
St. Louis area?
yeah, that I think they're spreading out, but in that general area, yeah. But the workers are morphologically identical. So I'm really curious as to how Antweb in particular deals with the classification of those when, they're getting, I mean, you wouldn't really be able to tell just based off of like a worker alone. You know what I'm saying?
Yeah, I think you would have to do measurements in this case. I think the measurements of the morphometrics differ, but it's not easy. And that's really gets down to this. It's easy to tell the cities apart, but now we're dealing with villages, and
Yeah.
it gets harder and harder. And to be honest, I've described species that I have to go back and try to go like, what? If I tell those apart again, go back to my work. I wrote the thing. I should know how to do this, but I have to go and do all the measurements myself again and figure out what species is this. It's challenging.
Yeah.
And I think there's various approaches, I think, going forward. It's true, an image. A lot of species can't tell what it is. Basically, you can identify any genus based on an image, which is pretty exciting.
Yeah.
And after that, the more information we have about existing distribution, the easier we can narrow down the possibilities of a current action. Of course, there's always new distribution records, but often when you're making identification, if you can have, example, right now for Colorado, you can say, okay, we know what ants are in Colorado. That's about the resolution we have. But if we had more information and knew that in this park, this is the list of species that are found there, and only those species, it would make identification so much easier. And so we have one level, the more you know about range distributions, the easier identification comes. And the more we've done the taxonomy, so we know that there are fewer and fewer undescribed species out there. So for the North America, there's no reason we don't already have both of those. You have so many collections, they're not available, so we don't have good range, even for the genera. I'm revising the Ants of North America book now, make it as large as the Africa and Madagascar book. And I'm just finishing the genus distributions, and some of them are really hard to do because I'm surprised we lack a lot of data in the Midwest. It's the kind of the Western Midwest.
I feel like, particularly in the United States, it seems like we have like pretty much everything you can find is described, but it seems that the descriptions in some cases are too broad. Like I caught Solenopsis molesta, I believe last year, and they had a late brood, and then when the Elatea closed, they started mating in the nest, and then the colony all of a sudden went from like 50 queens to like a ridiculous amount of queens. And so from my understanding, that's not a documented behavior with Solenopsis molesta, and so that's where it kind of seems like there's a lot of definitions like that where it's too broad almost. I don't know if you agree with that or not, but I'm just that's kind of what the feeling that I get from a lot of things that I see.
Agreed, but I would agree that there are still many species to describe. There's no way almost to identify gamma as a whole for California. There's many undescribed species, for example.
Yeah.
There's other groups during Myrmecology. You go through each genus, and there's always a limit in which you can have confidence to identify them. You can identify most Camponotus. Why don't we have great range distribution data? Why don't we have the taxonomy complete? It's maybe 10% of the fauna that still needs to be described. And why don't we have then a key to species? That's what we really need. Even so, we can dream that maybe in the future we can use what's called genetic barcoding to help with the identification. And if you're not familiar with that, that's taking a very simple PCR kind of old technique of amplifying one gene, what's called the mitochondrial CO1 gene, and using that. And it's also ripe with problems, but sometimes you miss the CO1 and you're getting actually another gene. It makes it confusing for the analysis. Or you've mixed up specimens, so you think you sequence one thing, but you actually sequence something else. It's 100%, but I think in the future, if you already had a species list for that park or that state, you would be able to actually take even an ant larva or ant male or a worker that looks just like all the other workers in Phytolii and sequence it, and you'll be able to identify the species.
Okay, wow, that would be pretty useful.
We could be there now, but we would need to have a little bit of focus. The background work needs to be done. We need to have finished a monograph on each of the genera of ants in North America. It's so doable.
Yeah, I was talking to. There was some guy up here who did a survey at one of our local parks, and I was like, I found something here. It was almost a decade after he did the survey, and it was like, we kind of hit a point where it's like, there's no way to really identify what I found without, looking at the genetics of it, because it's just the morphology is not pronounced enough for us. Maybe it's just us as hobbyists, right? But then, too, then it's like, okay, we're gonna go look at the genetics. How do I do that? There's no, I can't go on Amazon and order something to analyze the genetics of the ants I found. There's no resources for that. And so anything I think that we could do to move towards that would be a huge step for really the entire, entomology field.
Even if you had toolkit, and it's almost the point you could buy some portable ones that you just hook up into your laptop called Ion Torrent that people use now.
Oh wow, really? Okay, I had no idea.
There's only but they only work for a couple of samples, and you have to buy a new one.
Oh.
It's possible, right? So,
Yeah.
but that sequence alone is meaningless. You have to compare it to a known sequences,
that
right?
too.
The problem is a lot of people have sequenced ants in North America. Nobody's identified this.
Oh.
When you go and compare it to online resources, you're back to going well, who do I believe? It's this compared to that sequence. It says it's Dory myrmex. I compare it to that one. It says it's Lambotopum.
Yeah.
So that's why I say it needs to be a concerted, curated effort where you've sequenced identified specimens, or even the type specimens. Then for sure,
Mm-hmm.
But really has to be part of the taxonomic process of including these tools like barcoding in it, because the taxonomist has to verify that this specimen is that species before it can really become useful, especially for these cryptic ones like Stenema.
Yeah.
For Madagascar, for example, I thought we could do this, right? And I've sequenced actually 25,000 specimens from Madagascar.
Wow.
And it's becoming helpful because we've described now 50% of the species.
Yeah.
I mean, it's pretty helpful. But it's I think in the end, if we can finish describing, because as we go along, we vet specimens that we've sequenced.
Mm-hmm.
It's slowly building that.
Yeah.
Just alone, it's just oh yeah, there's all these clusters and what?
Yeah.
There's a lot of undescribed species, and we knew that before, so it hasn't really given us anything.
Yeah, wow, that's really interesting.
We talked a little
I also
bit.
I want to mention one other project though, which might be exciting to hear about, and that is called the Ants of the World. And it's a collaborative project that we are so lucky for the ant community to have funding from the National Science Foundation. It's in collaboration with a team of workers. Bonnie Blamer, who just moved to Germany, Berlin. Jack Longino at University of Utah. Phil Ward, UC Davis. Myself at Cal Academy. Michael Brandstetter and Logan. And we actually are trying to create the phylogeny for ants.
Mm-hmm.
A lot of people are working on this, but we want to move it ahead quicker. So we're sequencing about 5,000 different species throughout the ants. And this will build basically the first infrastructure for ant evolution. And it's going to be a real boom afterwards for scientific studies about evolution of characters, behaviors. And also it will help taxonomists, because these do taxonomy, like Dory myrmex, it's easier to first start and do the revision of a clade or a species group. You want to make sure you're working with a species group that's related. This taxonomic effort based on morphology is hard and slow. We've kind of wasted it, or by actually revising groups, not knowing at all what the phylogeny is or what the species groups are in any meaningful way. Camponotus. Camponotus has languished around the world because I don't know where to begin. We're sequencing them now, and we're going to have at the end, be able to say, okay, if you're going to start working on Camponotus of Africa, which hasn't been touched for 100 years, here's a group, do that group, because it's a clade, and start there. It's going to be incredible. I think it's going to be the most important thing. And second, we'll stabilize the classification, because at least we'll know all the genera after this, and the genera classification will stabilize for once. And you know how complicated it is to have the names changing all the times.
Yeah.
Me crazy. I have to rearrange the collection all the time.
Yeah, I know. I totally understand that.
And this will stabilize it, and I can't wait. This is one great practical benefit of this massive project.
Wow. So do you predict that in the next few years we're going to see maybe the number of genera double or even triple, or?
No, they won't double at all. They will change. What's well, a lot of groups, there's edge cases. So those edge cases, what genus does it belong to?
I see what you're saying now.
And they often don't just have to describe a new genus.
Mm-hmm.
They just get moved into the other genus. They were in the wrong genus.
Okay.
Then other times, like for Madagascar, it bothers me still. We know this, that Mesoponera is two distinct lineages. They're not even related to each other. We have two species in the Malagasy region, and they're completely unrelated. One of them belongs in Mesoponera, and the other belongs in another genus, but we don't know what it belongs to.
Mm,
We got to do the world phylogeny of Mesoponera to figure out how it sorts out.
yeah.
Same for other genera. The Phoenigaster in Madagascar is not related at all to Phoenigaster in Europe or North America. We still call it a Phoenigaster when we shouldn't be calling it a
Yeah.
Phoenigaster. So it's something else. We got to figure out what it is. So this is what I mean. And
I
so we
see.
know already there's actually three genera in Madagascar that are going to change their name. We don't. I just want it done, but we don't know where to change them to.
Yeah.
In other
Wow.
words, the key I just did still will change the future, and I don't want it to change. And having a stable generic classification allows us to have stable keys to genera. That's the entry point into the wonderful world of ants.
Mm.
Okay, I think Awesome Boss wants to ask a question now.
Yes, I do. First off, hello, Brian Fisher. I'm a very big fan. I've been admiring your work, and I've watched all videos about you with Ants Canada and all that stuff. So, hello.
Hello.
So, I have two questions, if that's okay. Um, First off, I am an ant keeper. I've been ant keeping for a few years now, and it's a lot of people call this it's very unethical. But what I like to do is I like to collect colonies from the ground, not collect queens during nuptial flights, but like during hibernation, I would crack open logs and or open under rocks and collect entire colonies. What do you think of that?
What I think it's the ant keeper movement is really driving new interest in nature, new interest in you can bring these amazing organisms into your house. It's, I think, much more exciting than having another plant or something. It's exciting. I think to me I see enormous amount of benefit for having people going out and collecting and bringing it into their house. But of course, there are ways to do it wrong and ways to do it right. And by the way, I just want to say that when I said earlier, I think we could all be scientists. I think having a live colony ignites excitement about scientific thinking, right? Because once you start watching a colony, you can't help but start. It just triggers questions, and once you start having those questions, I think you become more as a scientist, and that's great for society. It triggers so many things good about being human. It triggers hope. It triggers just this idea of being in harmony with nature. But that's a whole other subject. But let's get back to the practicality of what's the impact maybe on the environment. Now, of course, some places you're not allowed to collect live colonies. But there are many, many places where you can. Just going out into the forest, should you collect it or not? Well, you should be asking the same thing if you're bringing in you're hunting in a sense, right? So is it right to hunt deer? Would you classify hunting deer and a tiger on the same level, or a cat or other predator? No, they're quite different, right? So in the same in the ant world, you don't want to actually go out and kill a queen of an army ant, for example. That is completely unethical. You have to have a very good justification why you're doing that to benefit. Like I actually have. I brought a live 25,000 workers, actually more than that, 500,000 workers of army ants. I brought and put them on display. I had them on display for a year. At the end, I actually had to kill the queen. But I justified it because I was bringing it out for the education purposes. But killing a queen army ant, I would say that would be unethical, unjustified, just to have it as a pet, because it will die after a while. And I can't think of any other top predator like that, that would I would say you should not bring inside the house. Most temperate ants, if that's what you're thinking about, it's not a problem. You're not going to be impacting the overall uh, survival of that species or impacting the environment by removing just one ant colony. So that's my take on that.
Thank you. Um, my other question was, what do you think about the debate about keeping exotic ants or keeping native ants? Because I'm gonna be completely honest. Before I knew about the illegalness in the United States, I used to keep an Anachaetis colony. Sadly, they died. But and then now I realize the huge, the overall the cause of these exotic ants being imported to the United States or other countries. And I was wondering what you would think about like exotic or native ants. Like, what do you think about that?
I'll give you from my perspective. I'm a biologist, so I can analyze it in the point of view of impacts on biology. I'm not going to give you it whether or not it's a policeman would or somebody from EFA or USDA will have their own rules that they would judge this situation on. In terms of biology, is there a risk that that species will escape your house, become an invasive in North America? And that's going to be different. For a genus like Anacetus, there's a few introduced species around the world. Georgia doesn't think has any. There's Myrmecology in Florida. But it's pretty slim. For the kind of predators like the Ponerines, very limited. But I wouldn't suggest taking a Solenopsis species from South America and bringing it to the United States. I think that would not be a good idea. But most of the highly specialized, interesting ants pose no threat. If they would walk out your front door, they would not survive. If they're living in a tropical rainforest and then are presented in Georgia winter, they will die. That's from a biology point of view. In terms of like whether or not it has the label introduced or not, when you're watching it, it's still an ant. It's still amazing. That's from a biologist point of view. And of course, there's regulations about how ants are allowed to be transported and so forth. Those all have to be investigated. So could have brought that Anacetus from Florida or state line, and that could also break a law. But that's all those different rules have to be taken into consideration.
Thank you so much.
I love having cool ants.
I had a question specifically about like Proceratinae, the entire subfamily. I recently read in the Myrmecological news blog that there's a chance that Discothyrea could like preserve prey eggs using its saliva. Do you think there's any merit to that observation, or is it something that has to be explored further?
In fact, I don't know about that observation. So you're saying Discotheria can dissolve eggs using saliva?
Well, basically, I read a paper that it's an unpublished I was reading an article that is still an unpublished observation, but the ants will lick the species of Discothyrea was an Asian species. I think it's from Japan. I have to go check. But basically, they lick the prey eggs and then they turn purple or not purple. They turn orange. And it was brought up as a possibility that they might be able to preserve the egg somehow because they keep the eggs from like as an egg since the queens basically they establish themselves as after a nuptial flight inside of a spider's egg sac, and then they preserve those eggs until the workers hatch, and then even after that they can use those eggs for like several generations, which beyond the time that the eggs would hatch. Um, but my thing is, do you think they're actually preserving them with their saliva? Is it possible? Because like I don't know if it's like you know anything about it, but yeah.
Well, it sounds like that would be a cool invention. You think of it as it applying a of hormone onto the eggs that stops its development of some sort. So eggs are basically frozen in time. It's like maybe even imagine sticking a perilous venom in it, so it's just alive but not developing. So they've stopped development of the eggs, keeps them alive, and that's pretty cool. I'll have to ask my wife. She's an expert on Discotheria. She's been working on them last 3 or 4 years now, and I'll ask her if she's heard of this.
I put a link to the article that I found it in. Uh, it was an article in the Myrmecological news blog that I frequent often. It's in the Brian Fisher questions tab.
Okay, great. I just got there.
Um, and another question I had specifically for you was like, um, do you think it's was how much work is it to put in like on average to do a taxonomic revision? Like you mentioned, there's people doing taxonomic revisions on like Dory Myrmex, and that seems like it would be a lot more work than say a taxonomic revision on like uh let's see a species like I don't know Phaedoli where there's lots of I don't know like how much work would you say on average that kind of revision would require?
Well, revisions have steps. I think understand those steps. The best kind of article to read about how to do a taxonomic revision by Barry Bolton, 2007. It's a must read if you're thinking you want to start. Basically, steps you through the process. And I won't go into those steps now, but I'll say that the more specimens you have to study, the more described species you have to look at, the longer it takes. And the more specimens you look at, gives you more information about species. You want more specimens that you can describe the limits better. Not just specimens from one locality, but specimens throughout the range of the species. So if you look at Malagidris, that was maybe only took a month to do that revision because there were very few specimens in very distinct species. However, doing Camponotus now, revising that, and we have 6,000 specimens, over 150 species. It's taking a couple of years so far. Maybe it'll take 5 years. Phaedoli, another very difficult one. A lot of specimens from Madagascar. However, all the time you jump in after a revision. The easiest way to do first species is to pick a genus that was just revised, then find a new species in it. If that revision did all the hard work, and you got a key, and if you find something that doesn't key out, it might be a new species, and you already have all the background work done. A lot of people make a living out of waiting till somebody revises a genus, then they jump in for their country and go, oh, you didn't include my specimens that I didn't share with you, and I'm going to now revise them all. Unfortunate, when it was very standard, you did a revision, you would write everybody, hey, do you have the specimens of this genus? I'm revising it for the Palearctic region. And there was always one country that never sent specimens. And after they published that Palearctic revision, they would jump in and write their new species because all the hard work was done. That's another approach. But North America, there's a few genera that species, especially in Arizona, that you go down to and find a new species and describe it. You could
Ants.
do it. You mentioned already for California, there's even a couple of Camponotus. Those are pretty well defined. Those would be easier species to describe probably than Stenema. Sure.
Ants Ant YT wanted to ask you a question. I'm not sure, like, if he wants to ask it with his mic, he can go ahead. If not, uh, yeah, he can ping me. Hold on, I'm trying to find the question. I'll just ask it for him. Yeah, he says he cannot talk. So, do you think that Brachyponera chinensis is more spread out in the U.S. than what we think? Here in Wisconsin, we have a record, but it is away from all the others with. Oh wait, yeah, it is way all away from all the others with the same with Washington.
That isn't quite not aware of it being in Wisconsin. Check my maps. My Brachyponera map. I just did all these maps for North America. Might take me a moment to find it here. Maybe I'll do that for later. But yeah, it's possible. It could be anywhere. Anywhere you find. What's the upper limit for Brachyponera chinensis? How is the cold going to? At one point, it's going to drop out. They are up east coast a bit. What's in North Carolina, further north? I don't know the tolerance for that, but it seems like it could survive in Wisconsin, I bet. Yes, not sure. If you live in Wisconsin, you're going to be the expert on it. So track it down, figure out where it's found. Easy to identify.
I had another question. So basically, it's kind of about similar along the lines of like what determines different like casts, but in my question specifically, how much variation can happen like within a species, like especially coloration and anatomy before it's morphologically distinct enough to like be a different species. For example, I have two colonies now of what I think are Temnothorax regulatus, but the queens and the behavior don't match what was reported for the, and also the where it was collected don't match any of the observations reported like on AntWeb or on AntWiki. Because like if you go to the overview on AntWiki, they're always described as slow, and the species I found is super fast moving, and there's usually found under pinyon trees, but the species that I found was like under a pine tree, and one was in an acorn under a shrub oak. But they're the same. The workers and the queens look exactly the same, and the workers are like bicolored, but the queens are black. Um, but that doesn't match any of the descriptions for regulatus, which is the like similar, the most, and from what I have looked at, it's the most similar for both because uh, of like both of the things. But my question is, like, how do you morphologically discern like what, and can you behavior like if the behavior of one species uh, differs, or could it be the same species that has a differing behavior or something like that?
Could definitely be the same species and be showing different behaviors based on their history of a colony. Where they're at in their life history stages, the age of the colony, for example, determines their behaviors. Think of it this way, that within a genus, within a group of related species, there's often a gap in morphology between species, like head width and head length cluster. Ones form a cluster, a morphological cluster, and then there's a gap. There's another species that cluster on this grid of head and length and width. There's these gaps, and when you're trying to find out different species, you're trying to find limits of a species by finding those gaps. So there is always going to be variation within a species. variation often clusters, that there's a gap in that variation between species. That said, sometimes it's the behavior that highlights difference, and that requires careful look. Now, even an expert may not be able to tell the differences of the behavior just by watching them. They'll have to actually mount up a specimen, get it under a microscope, and do some measurements to figure out identification of those species. So you could also do that too by mounting them up and comparing the two species. You have to wait until maybe you want to sacrifice one individual from each one and look at it and study them and see if you see that they're different or the same. Then you use some of the keys, and I admit some of the keys are difficult, the ones by McKay and so forth, but it's what we have, so I think you should start there.
Okay. And another thing is like the species that aren't included in the keys, like the numbered species, like like for example, my examples with Tetramorium. There's like a lot of California Tetramorium that kind of match the issues I found with Rugulatus, but I don't know enough about them to know like what makes them different from the like how they were described differently than Rugulatus that makes them like
I think you meant temnothorax, right? Or
yeah,
leptothorax?
temnothorax. Yeah, it used to be leptothorax, I think, but now it's temnothorax.
Oh, right. That's the problem. It would be a lot easier to make identification if we already knew the limit for the number of species, but that's not the case. So it's difficult, and you would have to send it to the expert. And luckily, we have Phil Ward, who's thinking of working on that group, so you could send it to him, I guess, or Matt. What's his name? Matt, who did the revision for the Caribbean. He's also has an interest in this group.
Oh, okay. I have Phil Ward's email, but I don't know the other guy's information. I'll look it up.
He's at Arizona, working with.
I have a question. So, I noticed that in captivity, it tends to be that if your ants get some kind of parasitic mite or something along those lines, they tend to die out pretty quick. But obviously in the wild, that's not the case, right? Because all the ants aren't dying out. So, what do you think the disconnect is there? Is there it's just as simple as in the wild the mites have natural predators, or maybe you could shed some light on that?
Yeah, I think it's because they're not being contaminated. When they are, they just move. So ants move homes all the time. That's the great way to start afresh, is just to get up and move. Can reduce the parasite load to a point where they can control it. Then afterwards, a lot of those parasites are coming in from your feed, right? So
yeah,
they're in nature. They're not ever seeing such an unbalanced kind of environment where the feed they're eating is just already so so infested. And I think that's the number one challenge for keeping anything alive, like an insect, is dealing with other insects. It requires you to think cleanliness in a way that's mind-boggling. I mean, everything is a source of contamination. And we're talking about COVID all the time. You can't even go outside without thinking about how you're going to get yourself contaminated. Same thing if you're working in a molecular lab. Your DNA's everywhere. You can't work in a molecular lab without knowing where your DNA's going. Same thing with ant colonies. A mistake in the feed, and leave it open. Mites get blown around. They get into your feed. You didn't freeze it long enough before you used it. You didn't sterilize the sand before you used it. You didn't do this before you used it. It's a problem.
Yeah.
And you can wipe out colonies. Now, if it does happen, you got to take each ant out alone and move it.
Yeah.
To a place.
Do you typically see that with parasites? Is it pretty common to see them be cross species, or is it specialization typically with ants?
Well, when you see ants like with mites in the field, there's a whole bunch of mites attack ants and specialize on latching onto their tarsi or their legs or their heads. It's amazing. It's like they just specialize to hang on certain parts. So ants are a lot of species out there, but they never reach such an abundance as you find when you're inside your house. And those are not the same type of mites. Often a pest mite, and it's just a super annoying thing for managing their larvae. Their larvae are basically fast to death. That's the problem.
Yeah. Okay. Thank you.
When you're having ants, you have to be on the lookout for mites all the time. That's the number one thing to watch out for. Besides, things like not knowing what humidity ant needs. If you get it too wet, too wide, they just keel over and die. And not knowing what they eat. But besides that, if you have an ant alive, or know how to feed it, know it at the right temperature, you gotta watch. It can wipe out
Yeah.
everything.
Okay.
Flynn had a question, but he can't talk because it's like 1 a.m. I guess. So I'm gonna answer it for. I'm gonna ask him his question for him. I think his question was, have you ever kept ants as pets or in a lab? If so, what species and what was the coolest one that you have?
Oh, I used to have lots of cool ants. I had the advantage of at a museum. I already had an exhibit other ants. I actually was on the show, so we had army ants is one example, but we had a permanent exhibit of leafcutter ants. I had to have it a containment room was certified to bring in basically any ant species in the world, and I took advantage of it. Unfortunately, we remodeled our museum about 7 years ago, and we no longer had the space to keep my ants anymore. I basically moved all that kind of behavioral work to our collaborators in Paris, where it's very easy to bring ants. Europe there's no rules for this. But what I liked the best was I had most of the Amblypinae, Mystrium. I would bring back 40 colonies of Mystrium and feed them. Embryonid larvae, for example. Never had a problem with mites. Every time I brought in a grain-based species, I often had because of the mites got into their meat somehow. I liked slow-moving, beautiful ants. But they weren't actually great for show. When visitors come, they're like, that's it? I mean, moving. They look like they're frozen. So I always have some giant colony like a Phoenogaster or a honeypot ant people.
I specifically, I have two other questions. Um, uh, Basically, my first one is, like, I recently learned about like phoretic solenopsis and like ants that actually hitch right onto other ants. Um, are those like super rare? Because I've only like heard about them a few times. So um, like, have you ever encountered phoretic species, and like, why don't we see them more often than like we usually do?
Well, I think it's because as soon as you start collecting them, you don't see it because they all fall apart in your vial, and you're like, oh, look, I have a solenopsis and something else. You have to be very keen to watch them, and a lot of them are subterranean, right? So you're not going to see it underground. So I think it's I think it's really just by chance. And often, you have to be prepared to observe something. And sometimes when you just walk out in the field and go, oh, I'm going to go collect this Phoenogaster, just collecting the Phoenogaster, you're not coming out going, I'm prepared for a surprise. Let me watch. And so it takes a really special person, too, to be ready for a surprise, even though it's just a common species. Something to learn for that. Don't be a robot in the field. Be aware. Be alert.
Flynn's other question, which it was basically related to mine. I think you gave something in a TED talk about it. I read it on your website, but when you kind of talked about how you got the army ants to California Academy of Sciences, but like, where did you display them? Because when I went to California Academy of Sciences, like, did you put them in the actual outside of the dome, or were they in their like special setup or something like that? Like, how were they
Well,
displayed?
you went to our new building, which actually has a much smaller floor space, fun exhibits than our old building. And there, we basically turned over the entire public floor to an ant exhibit. It was so much fun. And we So we had displays of ant colonies and information about ants on the edges. In the center, we had a 6-foot wall glass wall that wove around into sections throughout the whole museum. The army ants ran through the whole thing. We had pillows set up on the floor so kids can also sit around it and watch it. So it was a vast area they could run, so they could migrate and move. So they could just move throughout the whole, basically, museum and keep going. But it had a very thick 6-foot wall all around it. And first week, they were crazy to get out of that exhibit. And I was going into the museum at like 1 in the morning with speeches that they would form this incredible ladder. They would be able to go against the 2-meter tall wall and stand on top of each other until they were able to clear the top. But at the top, there was actually a lip that an overhang of about foot that hung back into the nest. So they would climb up 2 meters and then fall backwards until they grabbed that overhang, and then they would crawl out. It was amazing they could do that. Those army ants were an amazing thing. They had all their commensals too, like all these staphylated beetles living with them still, and you could just watch them. I mean, it could have been the best place to film a National Geographic special. I really wish I was aware. Cool that it was all happening right there in San Francisco.
Another question I have also related to army ants is, like when you during maybe exploring the Southwest and stuff like that, I often hear about like there's a lot more like revisions on Neva myrmex, but I actually don't know enough about them as like where they're actually found. I've heard they're like subterranean, but like how do you like if I wanted to take a picture of them, what would I have to do to go and find them? Because apparently there's 4 species in Colorado, but I don't know how to find any of them.
Oh, you go out on a spring warm day, and it's even better if it's a hot summer night, actually. And you walk around some dirt roads, and hopefully crossing the road. They'll have a line crossing the road, and they'll be migrating from one bivouac to a new bivouac, and they'll be just migrating. I mean, I do see them in the Bay Area, but not very often. But I do see them much easier in the summer, like in August in southern Arizona. They're much easier to see. You can predictably go out and find them there.
Let's see. I'm trying to see if there's any more questions we might have missed. I think there's a few. Let's see. If you could take 10 items on a collection trip, what would you bring? Only 10. You could only bring 10, and what would they be?
Well, it's not just like 10 vials. I could bring vials, but I would need a GPS. I would need vials. I would need my little machete digging tool combo that I have. And I would definitely bring my magnifying glass because I like to be able to identify what I'm collecting immediately. It's a little eye loop. That's essential. Is that 4 or 5? Close. And then I need a notebook to keep notes in. And I would bring my leaf litter extracting equipment because if my time is short, even though I love better just watching and looking for ants, if I want to make sure I've got a good sampling, I'll take some leaf litter samples because that's the quickest, most effective way to capture diversity in a forest habitat, rainforest or any habitat. Nothing beats leaf litter sampling. If you only have an hour, you can still capture a lot of interesting ants. If the leaf litter is a bit moist, if it's super dry, then it's hard to find ants no matter what you do.
One of the more like questions, more like ones I probably should have asked earlier is how has the COVID-19 situation affected your daily job and activities?
Oh my goodness! Last month, March, was in the middle of a beautiful rainforest, some of the only lowland forest in Madagascar. We were actually hunkered down because a cyclone came, Harold cyclone, that came from Union back into Madagascar, and we were rained on 5 days straight. Never seen so much rain. About a meter of rain fell in 5 days. So much high wind, maybe about 100 kilometers an hour, but the rain just would not let up. Then during this, we get word that the country of Madagascar was shutting down, and the last plane out was on March 19th. So we actually did make it back to the capital on March 19th and flew out that night. And since then, I've been home writing papers. I haven't been back to my office since basically the end of February. Who knows when I'll be back. And this is another important point. It's all fun and games until you find out that museums are public institutions. Our museum is a private institution. Our museum requires visitors. So, and we're going to find out what that means. Most museums have already cut their budgets by 30%. And that could happen to ours too.
Two more questions, I think. One I wanted to know is how important is learning a second language in an entomology or biology degree in general?
It really is helpful. When I decided I was going to be a tropical plant biologist, I started thinking about learning Spanish and French. I'm glad I did. Those two languages allow me to work most other places, and I picked up most Portuguese too. So I can go almost in any tropical country and work, and it's great. I think makes it easier.
Awesome, boss.
So you can be strategic. I'm saying you can figure out what you want to do and go for it.
Awesome Boss says he has another question to ask.
Hello again. Yes, I do have another question. So in one of my colonies, I have a few subterranean ants. I have this Solenopsis carolensis 3-queen colony. Sorry, something happened. I was wondering, I had a question. My bad. I was wondering, like for some cryptic species like Molesta and carolensis who, feed on eggs, why do they like feed on eggs? What is like, is it discovered why they just feed on eggs and not on other insects, or do they feed on insects? I don't really know about that. It I have researched about it, but it's not really discovered. So if you had an answer, that would be great.
You're Carolinensis, right? And Molesta? Like the Solenopsis thief ants, I think you're talking about.
Yeah,
Yes.
those.
Okay. Yeah, just to clarify.
So eggs are, an incredible resource for rich, purified with protein and micronutrients. It's a gold mine, but very small. So you got to be a small ant to benefit from it. And small allows you to sneak in, get it. So we think of the soil as a solid substance, but it's actually a mosaic of channels and pores, and Onopsis molestus-sized ants are perfect to just kind of be like liquid through that habitat, and it's incredible. It's hard for us to imagine, but they're in there. They're super tiny. And ants provide an incredible pocket of resource in the soil as eggs and as larvae. And there are, all the army ants, all of them eat ant larvae. It's just incredible to think that it's enough in North America to have a whole group of ants just specialized on eating them. And it's also, they're another way to get by is I just only hesitate because I think that some Solenopsis will take the larva too, so it's maybe not just eggs. You can't feed your colony eggs, so what are you feeding it? You're probably also giving it some fruit, maybe some grapes, so they get some moisture. Maybe I'm sure will also eat, if you slice it and you beyond a larva up, they'll go in there. I'm curious, what are you feeding your Solenopsis?
I don't know about him, but like in Colorado, they're fairly easy to keep because they'll pretty much eat anything. They just prefer to eat eggs in the wild because it's an easy, accessible food source. But basically, they'll eat fruit flies. I've seen them eat mealworms. One time I fed mine a dead fish. Um, and then they'll eat all sorts of liquids. They like most ants. You feed them honey and they'll drink it. Same with beetle jelly. Beetle jelly is my like number one thing to feed ants, especially smaller ants, because it's more viscous than honey. And even if the ants, like the cool thing about Solenopsis is like Monomorium's just a bit bigger than Solenopsis, and sometimes they drown in honey. But since Solenopsis is so small, they don't drown in honey. But regardless of the size, if you feed beetle jelly to ants, it's more viscous. Um, so like, and sometimes it has protein in it, so you can feed a colony entirely on the beetle jelly. But yeah, they'll eat that. I've pretty much gotten them to eat anything. Although I don't really know like what species of Solenopsis is in Colorado because most people say it's Molesta, but after looking at Wheeler's like 1917 paper about the mountain ants of North America, and then I went and go check the Solenopsis molesta castanea, which was reclassified to carolinensis. Apparently, I'd been misidentifying all the species like for almost 2 years, but um, like I think I sent you a picture one time, but you know that queen with the eye, like the larger eye.
Right? Yeah.
Um, well, when I looked at it, it actually made sense because uh, the carolinensis, like if you take into account the molesta castanea stuff, the 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 thing is that most of the identifications could have been of like carolinensis because it's a darker species of like a darker variant of that species. So it was
And
interesting to find out.
I have something else to add on to that, if that's okay. Um, I recently collected my Solenopsis carolinensis colony around 2 to 3 days ago, and I have fed them mealworm and honey. Um, I don't think they haven't reacted to it well. They haven't. I don't think they've drunk the honey. I don't know about the mealworm. I haven't checked on them, but I'm not so sure. Um, Maybe it's a possibility that they're right now eating it, so I'll go check on them later on. But if they do eat insects, it'll be a lot more easier for me to feed them.
Well, usually they're just so small that they take forever to eat food. That's just from my experience. Same thing with like Temnothorax and stuff like that. Hey, Manitobant, if you have any
Hello?
questions, you can go ahead and ask them.
Okay, I wanted to ask, oh, are you listening?
yes,
Yeah, so I wanted to ask, what are the requirements that you need in high school to become a myrmecologist? Like, what courses do you need?
I think high school is a great time to explore. Where you can create free time, in a sense, to explore ants, the world of ants, and you can look at it. It's kind of learning your skills for collecting, identification, and keeping. Um,
Yeah.
it and also think of creative ways to be, find new ways to keep ants. And each species has its own lifestyle and its own footprint in the world, and you may have to invent a new nest structure. Learning to watch them. Becoming an expert on ants is really helpful when you want to do research on ants. A lot of people come into the ant world because they're interested in science. So when they go to grad school, they're like, oh, I'm just going to start studying. So most of the scientists out there doing ants don't have any background at all. Don't have that kind of depth their questions to really be understanding ants. Because that takes time, and there's no time for that in grad school, ironically.
Oh, so you
They're,
don't need, like, math or science, or do you need
but
any
you
of that?
will, yeah, and that's all you're going to be doing then is math and science. So do science, you're going to need math and programming, and basic entomology skills. And being a undergraduate is the best place. But of course, when you're in high school, make sure you keep up with math. And if you can pick up a programming, Python, you can learn so much quicker when you're in high school. Also, it's the time to have, basically, once you start college, you have less and less time to do something as when you start studying ants, you can do it without a clear objective. You have time to explore and do things in high school. As soon as you get to, undergraduate, it gets tighter and tighter in terms of you have to make sure what you're doing will result in something. In graduate school, it's even worse. You don't have 2 years just to kind of play around and see if you can find something. You go to grad school, you already have to know what you want to do in a sense. So take advantage of high school to really, not just ants, but ants, are part of Hymenoptera. Learn something about ants in general and Hymenoptera. You can't be a scientist also if you don't start learning math and programming and biology. You got to do that too. I'm just saying that, take advantage of these clubs, take advantage of the fact that you have weekends where somebody's not saying you have to go to get a job and make money, and explore the world of ants.
Okay. I know I want to study slave raiders and social parasites. That's what I know I want to do.
You already know, there's 3 people in North America that are great to do that. And who they are?
Who?
Okay, I didn't know if you might have already corresponded with them, but are you in North America? Where are you based?
I'm in Canada.
In Canada? Well, that's still, there's some Canadian you can work with, but the closest one then, if you want to go Canadian, slave ants in the group Formica, be Myrmecology.
I know who Popoff was. He did Polyergus in Arizona before he died.
Yeah, Howard was wonderful. He was one of my guest lecturers on ant course there. Now we have other people studying Polyergus, which is Neil Tetsui at UC Berkeley. And all of them take volunteers in the summer if you wanted to go study with him. Neil Tetsui at UC Berkeley, or go to Montana and work with...
you can do that?
Yeah, people do all the time. I've had loads of high school students to my lab. They're usually the furthest away is from California or something like that.
And I just I can just if I sign up for Entomology Society of America or something like that, I can find them and stuff like that.
Usually in science, it's all specialized. So you write somebody and say, "Hey, I have a big interest in this, and I'm looking for any opportunities to do an internship at all. What can I do? How can I get involved?"
Thank you.
And there's also one in Arizona too. I just wanted to mention the third one working on parasitism is Abilene, University of Arizona, at Arizona State University in Tempe.
Oh yeah.
I had another question, about specifically your lab and so when you do you ever do I know you do the intro to entomology at I think UC Berkeley, but do you ever do short introduction to ant classes or stuff like that at your lab for for example, if I wanted to learn how to properly pin ants, there's a museum here, but they don't really do much Hymenoptera. Uh, I'm working on probably expanding their collection this summer. But if I wanted to learn more about ant collection techniques, do you I know you do ant course and stuff like that, but do you do a domestic lab course? I know you mentioned that, but how do I learn more about that?
I can just tell you about what other high school level students have done. It's only difference, say high school versus undergraduate and up, because undergraduates and up can come alone. High school have always sent, it's often their father and a son, or the father and their daughter. They'll come to San Francisco for a week, and their son or daughter will be in my lab learning things. Right, the same things happen for the course that I teach in Sonoma, which is just an hour north of San Francisco, where they'll stay at a hotel and their son or daughter will take the course. It's only
Oh,
because
yeah.
it's a 3 day, it's a weekend course, so it's only 2 days. And so they spend one night away from their child. And of course, there's all these liability issues that the parents have to think about. And like, well, should I have my kid? These group of college students are not in my lab. It's the parents are right there. So, um, and very dedicated parents that show up and sit there while their interested student, son or daughter, have sat and gone through workshops. And one who is close, like in Santa Cruz, they come up once a week for years, work in the lab.
So, like, for
Learning to mount.
example, like I live in Colorado, but if I wanted to do something like that, would it be open to me if I like flew in and like went and visited for a course or something like that? Because my parents are definitely okay with that kind of thing. Because I'm doing it next year for Bugshot, definitely, and other trying to do other stuff like that. But is it have
Yes.
you yeah have you ever done it with people outside of the state of California, or is it usually just people in California?
Uh, I don't know.
Cause like if my parents were just staying in a hotel for a couple of days, like we have like
Well,
my
if your parents come, it's not a problem at all.
yeah, okay, because they would probably come and it would probably be my mom because I used to live in California when I was like really little, but they lived there most of their lives, so they have like all sorts of friends and people in the Bay Area and stuff like that that we can go to their house and stuff like that.
To be able to do it the first time, I had because we are a public museum, I had to be certified to be with uh, underaged children. I didn't know there was such a thing. Guess you have to do that in high school to be a teacher, maybe. But our lab is certified. We've all been
Yeah,
asked
when I
all our courses to be able to handle underage children in our lab, so we can do it.
yeah, when I talked to Dr. Krell, who he's the curator at the museum in Denver, he said they do like a teen internship thing. It's general entomology, which apparently they're still doing it this year, which I'm surprised that they're trying to still do it even though they're not allowed in the collections yet. But anyways, he said one of the main issues, like the same thing as liability and stuff like that, and he had to start separating different like he had to space out the internees because you can't they have to have a certain variation in the age group. It can't be too large or something like that. So he had to start adding limits and stuff. Uh, but anyways, the reason that relates is do you think that if there's an entomologist, like, so for example, if someone doesn't have parents who will go with them to California while they take a course for 2 days, because I know a lot of people, their parents aren't open to that or can't afford it or something like that. If there's a local museum that teaches you entomology, like, or does an internship or something like that, uh, are things like that good for learning how to pin ants? Like, for example, if you're pinning a small beetle, is that usually the same process as pinning an ant? Like, if you point mounts a beetle?
It's not the same process, and you'll still learn how to handle pins and labels and stuff, so it's good. I won't teach you the specifics of ants. I really listening to you, and it reminds me that I need to create these full two videos. It seems really like a missing link in the ant keeper community. I think now that it's growing, it becomes more important. Before, I directed those videos mostly at future ant taxonomists or ant students, but I think it needs to be much broader now and very, very simple. And luckily, most of this equipment is accessible, and that's important. Out of the reach of a lot of people to buy a few pens and a container, the ants in. So I think it's doable.
Okay.
Okay. I have a question.
Uh, Before we do your question, Manitoban, Echo Meter, he wanted to
Oh,
ask a
sorry.
question for a while,
Oh,
so
I'm so sorry. I'm so sorry.
yeah, we
Yeah,
can do you next.
okay. Well, for keeping ants, obviously, you need the ants. So I would like to ask for like collection methods. What are like the most used to get colonies that have queens and all of that, and also like the specific ones, like for example for twig ants and all of those different ants that nest in different spaces.
That's such an interesting question. I had amateur join me. He was actually a student of the course. He was an amateur, but I was able to get him into the course. We taught an ant course in Ghana, a baby ant course last year, not the official ant course. And he came along, and he's been collecting ants for like 15 years. His name is Gico Gomez. You know him. He's an amateur in Spain. He's has a regular job, but just collects ants on the weekends. Anyway, that was like the number one thing that just shook him. He went out in the field with us and watched how we collected, and he goes, "Oh my gosh!" First day, he came back with Phydole and Camponotus. Like, oh, is it? And we came back with like 50 different genera of all these things. And so you're right. There's something you have to learn to sniff out the ants, and you have to realize where you'll find the most ants or where you'll find the ant that you want to collect. And for example, if you're in a forest habitat, often in rotten wood structures, is a great place to find ants. And there's a seasonality to it too. Some seasons are easier. Springtime, when the ants are more active, when you can find smaller colonies, that's maybe the best time to go out and collect. And you have to be in a place where you can be a bit destructive. You can't do it like where all the tourists go or hikers go and they see you that you're going to dig into these logs. You got to get off trail. You have to go into the forest. You have to create a mess in a sense. But you're a good ant collector and kind of clean it all out when you leave. You're going to turn over 100 rocks. Make sure when you come back, you've turned over those 100 rocks and put them all back. You don't want to have ant keepers a bad name by leaving a trail of destruction. Be part of the good etiquette to learn, but you do have to be destructive. You got to put it back. You have to dig holes. You have to turn over lots of rocks. You have to turn over branches and break them open. Just make them kind of neat when you leave. That's how you're going to find those ants. You're not going to find them just walking around looking for the ants walking around. You go where they live, dig for them, look for them. It's always amazing. You go and climb a tree. We teach a canopy course for undergraduates in Madagascar, actually. Supposed to be in June this year, but we've postponed it now to January. Hopefully in January we can teach this course. We teach students to collect ants in the canopy. It's funny. First time they go up, they go, "We're in the canopy. Where are the ants?" They were kind of expecting the ants to be everywhere, but it's the same thing as on the ground. You're not going to find any ants living in the trees until you start breaking branches. That's what you have to learn to do, is guess where the ants may be and start breaking the branches and looking for that type of dried twig that's just the right size, that genus.
Wait. Okay. So wait, was he done? Sorry.
I'm done. If you have more questions, I can answer them.
Okay.
I wanted to just do like a little extension to that question. Just um about traps. If there are like some commonly used traps that work effectively to luring colonies or something like that. That's all.
For traps, they're very effective, but you won't get the colony. You'll get the individual. So like pitfall traps are great in certain habitats like woodlands. You get leaf litter sifting will give you grass, like in the forest. Leaf litter sifting is great. They just give you records of the presence of the species. They give you hints about where you should be looking. So you want to know what's in the canopy, you can set out some baits up in the canopy. If you collect an ant that you want to collect, you know it's nesting somewhere in the canopy. It's not going to tell you where you're going to get that colony. You're going to have to look for the colony. That's different. Traps will not give you a colony.
All right, Manitoban, how about you ask your question now?
Okay. I actually have a few questions. First of all, I'd like to ask. First of all, I'm kind of interested in doing this ant course stuff and like these meeting with seeing the lab and stuff. But the thing is, where do I contact you guys? Like I've seen Entomological Society of America. That's what my friend told me. But like, where? How am
It's
I?
you have
Yeah,
to
like
find the email of the researcher you want to contact, and then email them.
oh, you just like go like my friend told me to check Entomological Society of America or something like that.
Um,
Maybe I'm not familiar with that in terms of an index of names and addresses, but me, you just Google their name and find their lab and look for their email address. And if you can't find it there, you go to the most recent publication. And in our publications, we always list our email addresses.
Because
also,
Then you
like
email, and
it,
when you
I'm
email them, you have to be very clear why you're emailing them. So introduce yourself as somebody that's just getting into ants, or you've been studying ants yourself for a few years, have this particular interest in, for example, making ants or whatever, and you're looking how to a way to advance your interest and interest in science. And are there any opportunities with you to do that in the summer
okay.
for an internship?
Because I'm also interested in army ants as well, like Niva Myrmex, Dorulus. I know one researcher there. I think Gordon Snelling. Doesn't he do Niva Myrmex?
Well, that's true, but he's like an amateur, right? He's works in a pesticide company, but does some ant work on the side. It was his father, Roy Snelling, that was the ant expert, but unfortunately he died on an expedition to Kenya years ago. But he did teach his son, Gordon, a few things. But there is a very, very interesting researcher, army ants, based in New York. Uh, would be at the Rockefeller Center. Be sure. He has a new book on army ants. You should buy his book and read that, and go work with him. He's a wonderful researcher.
And you can find a list of these names where like and stuff like.
um,
Sorry.
well, if you want Brian Fisher's email, I pinned it in the questions. Um, but as far as other people's emails, uh, if you can't find them in their publications, which you usually can, uh, if you can't find them or you can't copy them because of the way the PDF works, you can always go and try and look them up on AntWiki because that usually has their email and sometimes their fax and other ways that you can contact them. Uh, let's see. And then also just looking the person up sometimes brings up their and visiting their website too. But
Okay.
um,
And
yeah, go ahead.
oh, and my other question was so it's something I was wondering. So first of all, I'd like to give some background. I have Asperger's syndrome, and I was wondering, is that going to inhibit me from becoming a myrmecologist or something like that?
Well, we already have a few, um, and so I don't think that'll be a problem. In fact, interesting enough that one of the students we had that came to our lab learning before high school at 9 years of age also had Asperger, and his father was just happy that he had he had an interest that could be developed. This student, unfortunately, was very talented in the programming side. Went out the ant sense he studied robotics at Stanford
Oh
instead
yeah.
of studying robotics in the traditional way. Tana took a multi-robotic approach, which then all of a sudden became very fashionable. So he hit big in the ant world in a sense.
Oh yeah.
Just hang
Um,
on. Anna, phone call. Head.
well, let's see. I'm trying to see.
I have a question, if you guys don't mind.
Yeah, I was gonna say if anyone else has questions, go ahead.
So in the society of all the ant keepers and all that stuff, um, when you usually keep an ant colony, it's usually for two reasons. One, it's for keeping to have fun, and two, for selling purposes and just to make quick money out of them. Um, a lot of people do you think it's ethical to just keep ants and to buy them from other places just to sell them. What do you think about that?
Um, Everything has a value. If you had a really cool ant I was looking for, I hesitate to buy it from you. No, I would buy it from you. It has a value to me. It'd be much cheaper to buy it from you than actually go there and find it. And that's a real thing. I got a chat message once that somebody was selling an ant. They didn't know what it was, but it ended up being Apomyrma in southern China. I just missed buying it. That person actually who bought it will be revealed to me, but I missed an incredible chance. It was the first queen of that ant genus ever recorded, first larva ever recorded. Somebody else eventually found it and got it to a scientist, and they published a paper last year on it. But I missed an opportunity, but I would have bought it. It was $50. It cost me thousands of dollars to go to China and try to find it myself. But that's just saying that even I would pay for it to do science. Would you sell it just to have it? But you're selling it to somebody who's going to actually put it as a pet in a sense, and they're going to be with nature and they're going to observe. And that makes them, I think, a better person. And they're providing a service. Now, I think you have to ask, are you having an impact on the environment? That's the real question. So, no, I don't want you selling army ant. I would just get out a top predator from the environment that requires a huge range of land to actually live. That's wrong. But most of the other ants, it's not a problem. Thousands and thousands of eggs are produced and things are produced that are lost in the environment. So I don't think there's an issue. I don't think there's a biological point. People have an ethical point because you shouldn't sell living organisms, but we do it all the time. We buy cows, we buy meat. So, to be honest, I don't think it's a problem.
Oh, okay, thank you. I have one more question if you don't mind. So when you want to become like a myrmecologist and you want to go to like a school, like a school of your choice, is there a way to get a scholarship for myrmecology, and like is there a way to get credits through myrmecology when you're undergraduate?
I don't think there's a way to get a particular scholarship as an undergraduate, but as a graduate school, in a sense, you don't pay for graduate school when you're going into myrmecology. When you're going to graduate school, you're accepted into a program. They're offering you ways to pay for it through helping to teach, doing research, or through scholarship, fellowship. Yes, at graduate school, you want to be a good myrmecologist, so you get a good offer to go to school. And that requires planning from the very beginning, right? Learning some language, learning some computer skills, and experience, maybe even publishing a paper before you start graduate school. That's why it's great to start all this in high school. As an undergraduate, though, there's no way to get a scholarship that I know of in myrmecology, for example. You could take a course like ant course as an undergraduate. We give out scholarships for the course, and we've done that to a lot of undergraduates. So at that level, you can get scholarships.
Another college-related question. Basically, I've heard many ways, and from many different opinions. But as far as college goes, do you think it's important to do your master's degree, or do you think if you just skip the master's and go to a PhD, it's it's not as important to do the master's? Because I know that people feel differently about that question, and I've asked it to multiple people.
Well, they have two completely different roles. It depends on the person. So there's an interplay between the person and the institution you're at. It's a good match. Why do you want to do a PhD in the first place? It's to give you the credentials, open the whole world of science for you. Once you have a PhD, you can apply for your own funding, a job, you can spend the rest of your life doing science. You need the PhD. If your goal in life is to go that career, you need to get a PhD. There's no reason to do a master's if that's your goal. However, there's a lot of other reasons to do a PhD or to get experience. Your goal may not be long-term career in science, but you need the background to go on and get a job in land management or conservation or any other aspect of entomology. A master's may just be sufficient for that goal. And it may be the case that you're not ready to start a PhD. You haven't figured out what's your main interest. Or you go to an institution and realize, you don't like my advisor. Maybe a master's is better. So masters have a role, even if you want to go on and be a scientist. It's when you're not ready yet, think yourself into 4 years of one topic, or you haven't figured out what your topic is, or you're not, you think you need to figure out if you're ready for it or not. If you're going to go on to a career, you don't want to spend time doing a master's, a PhD, because that opens up the world for you. And you want to get that out of the way as fast as possible. Spend 10 years doing it.
Um,
Okay. Oh,
Go ahead.
he can go. He can go.
Well, I was gonna say, yeah, thank you for that. I've gotten a lot of answers on that. Most of them have been saying it depends on like what you want to do. Like I heard someone say like if you want to work at a government organization, I guess you can't do that if you have a PhD. But is that true or do you know about that? Like for example, if I got a PhD in myrmecology, would I be overqualified to work like at on the state level on ants or like
No,
is
not at all.
okay.
Be overqualified.
Yeah, okay. All right. Manitoban, you can go ahead and ask your question now.
Okay. I wanted to ask, when I email these, I'm sorry I keep asking about this, but like when I email these like myrmecologists, do they often accept young high schoolers?
People have summer programs, used to having visitors and internships. Whether or not, next summer they're available, that'll depend on that person. He could be on sabbatical, it could be something else going on, he's moving. They have to, writing, getting a conversation going.
Because I'm thinking of
Science is a
contacting.
community. It's
I'm
not a
thinking of
yes or no answer. It'll be depends.
Yeah, because I'm thinking of contacting like the guy that you said about slave raiders and maybe going with him for like the summer.
Right? And to be honest, when you say they're going to go to that lab, you're going to the lab. That means you'll be working with his PhD grad students or postdocs who are working on the project, which is better. Professors like myself hardly have any time, quality time every day for 8 hours a day to work with an undergrad or a school students, but the lab will help you. So think of it as the
Oh,
lab.
where do I go to like work in the field if that's possible?
You can volunteer for somebody in the summer, I guess.
Oh, I'll try going in the lab then.
Um, I had a question specifically about Proceratinae. Do you know if anyone specializes on the New World uh, application of that of discothyria and Proceratium? Because it'd be interesting to look at that kind of thing because haven't been able to find like a specialist on that or someone who's done work on them in the past.
No, nobody's really done any work in the New World on it.
So is that like if I wanted to work on those, maybe like if I decided I was interested in learning more about them, it would be pretty much uh, almost all of the observations would be newish.
Yes, definitely.
Because because of that one article I read, they're now kind of like the most interesting ants in my eyes because they're really tiny ants and they're round, and apparently they might be able to preserve prey eggs, which that could have applications in like food preservation technology or something like that.
That could be really exciting. They're tiny, they're hard to see, but it would take somebody like you to actually have the time to actually watch them. I just want to give you a heads up. I'm running out of time.
Yeah.
Jump to another meeting.
Okay. Well, we're super thankful for having you, and I just wanted to let you know, like, if you do end up, wanting to make those videos, in the future, like, part of our club effort is to kind of get some educational content out there. So, like, if you ever like make those videos, let me know so that I can post them in like my content channel so that people can look at them and know how to collect ants and how to pin ants and things like that. Um, but other than that, it's been such a wonderful time. Thanks for coming. Uh,
Yeah.
does anyone else have questions?
Yes. I just have one more thing to say. If we have any more questions, can we email you?
You can, and sometimes I can email you back. Sometimes I wake up in the morning and find 100 emails and um, miss a few, but most of the times I'm able to do it.
Okay. Thank you so much.
And I love the fact that you guys have formed this group and all interested enough to learn more. And they'll have to ask you about why you have so many strange names, but maybe next time about your names here.
Yeah. Um. And if you're ever just like if you want, we can keep the Brian Fisher questions open, and you can come check it every now and then, and if you want, maybe answer a few of them. But if not, that's cool too. Um, anyways, thanks for doing this with us. Uh, I know a lot of these people were super excited to get to listen to you talk. Um,
I was honestly super nervous.
yeah.
There's no reason to be nervous. All
All
right,
right.
everybody. All the best to you, and
Have a
we
good
all
rest of
stay
your
safe.
day.
Yes.
Bye.
All
Bye,
right.
Dr. Fisher.
Bye.
Bye.
All right. So everyone else, I guess that wraps that up. But if you have anything to discuss or talk about, or you want some information, let me know. I am going to take that and make it into a video. If anyone else recorded that, please let me know because it's my first time doing it. So, you know, uh, DM me some like if you have if you have any recordings or something from that.
That was
But
a lot more than I thought it would be.
well, yeah, I thought it would only be 45 minutes because he was like, "Hey, can we restrict this to 45 minutes?" Well,
Yeah, same.
it turned out like, hold on, let's see, how long was I recording? 2 hours and 27 minutes.